The document that I'm going to be scribbling in the video would be provided through a link in the description, and also the link would be there in the first comment. Please do check it out, and let's get on with the video. So there's a system in the prokaryotes or in the bacteria which is called the Operon system. Now, if we define the operon, then an operon would be a group of genes under the control of a single promoter. And also under the control of a single regulatory unit, and also a single regulatory unit. So this is the definition of operon. What I mean by this is, if let's say I have three genes A, B and C, then all of these three genes would be under the control of a single promoter, let's say P. And promoter is the place where the RNA polymerase binds and starts its transcription. And also, apart from the promoter, there is also another element on the DNA which is called the operator element. Now, like on promoter, the RNA polymerase binds. On the operator element, the regulatory protein would bind, and it would regulate the expression of the genes that are present in the operon. So these genes, these are basically called the structural genes. Right, so this is the the overview of what is an operon. Now, over here we are specifically talking about the Lac Operon. That is, we are talking about the lactose metabolizing operon, the system that is metabolizing lactose inside a prokaryote or a bacteria. Now, lactose is made of glucose and galactose. So lactose is basically a disaccharide, so it is made up of two different monosaccharide units. Say, for example, if this is a culture plate and you're growing bacteria on this culture plate, and you provide as carbon source both lactose and glucose on this culture plate. Now, whenever there is glucose present for the carbon source, the bacteria would always utilize glucose first rather than any other sugar molecules that are present. So glucose would be utilized first, followed by the other sugar molecule that is present, in this case, which is lactose. So there would be a graph. If we plot this onto a graph, then let's say this is time and this is number of organisms, number of bacteria that are growing over time, we would get the curve like this, which would eventually flatten out, but it won't go to depth. So this curve would denote the growth which is done through utilizing glucose. Now, when it was about to go to the death, that is, when it was about to decline, over here, then it would eventually know that another type of sugar, which is lactose, which is present in the culture medium, and eventually it is going to take up the growth from here, and again it is going to flatten out. So this curve, the second part, it is because of the lactose which is present. So altogether, we call this kind of growth curve, which is seen in presence of glucose and another sugar, as dioxic growth curve. So there would be two different curves, one for glucose and one for lactose in the same medium. Now moving on to the Lac Operon. So the Lac Operon, like I said, it is specific for metabolizing lactose, which is present in the medium. So the Lac Operon consists of 3 different structural genes: the LAG-Z, the LAC-Y, and the LAC-A. And like I said, we are going to have a promoter element upstream of the whole structural genes. So let's denote this as 5' end, let's denote this as 3' end. And apart from the promoter element, we are going to have the operator element, right? So LAC-Z, it is responsible for encoding a protein or an enzyme which is called beta-galactosidase. LAC-Y is encoding a protein which is called Lac-permease. And LAC-A is encoding a protein which is called lactose or acetyltransferase. So these are the 3 structural genes that are encoded by the Lac Operon. Now, beta-galactosidase, what it does, let's talk a little bit about the function. Now, beta-galactosidase, I'm writing it as beta-gal. Now, beta-galactosidase, it cleaves the beta-1,4 linkage or beta-1,4 glycosidic linkage between glucose and galactose present in lactose. Right? So like I said, lactose is made of glucose and galactose. So in order to utilize glucose, so they need to be broken down into the 2 components, that is glucose and galactose, and then it can utilize glucose. So it is breaking the bond between glucose and galactose. So that is the work of beta-galactosidase. Now if I talk about Lac-permease, what it does, it encodes a protein which embeds into the plasma membrane and allows entry of lactose. So whatever the protein is being formed, they are being utilized in the metabolism of lactose. So Lac-permease, it is basically, if I draw it over here, if this is the plasma membrane, then Lac-permease, it sort of creates a pore on the plasma membrane, which allows, if this is the outside and this is the inside, then it will allow lactose to come inside the cell. So it is permitting the entry of the lactose. And transacetylase, which is not so important, transacetylase, it is responsible for adding an acetyl group to beta galactosidase. Right. So these are the different functions of the different structural genes that are present in the Lac Operon.